Good day everyone, welcome back to our channel. We are about to do another video on KREB cycle. Do well to like, comment and share. At the end of this video, we are supposed to know the reaction of the TCA cycle, the definition of the TCA cycle, the irreversible steps of the TCA cycle, the oxidation reactions of the TCA cycle, the oxidative decarboxylation reactions of the TCA cycle, and other things. Now, so let's define the Krebs cycle. The Krebs cycle is an anabolic pathway that oxidizes acetyl-CoA from carbohydrates, proteins, and lipids. This carbon dioxide, ATP, and high energy carriers like NADH and FADH2. Krebs cycle is also called TCA cycle. We can also call it CAC cycle, which is citric acid cycle. Meanwhile, it's called Krebs cycle because it was discovered by a man called Hans Krebs in 1937. It's called TCA cycle. TCA cycle stands for tricarboxylic acid cycle. It is called TCA cycle because some of the molecules in that pathway contains 3 molecules of carboxylic acid. It is called CAC cycle. CAC cycle stands for citric acid cycle. It is called CAC cycle because the first product of that cycle is citrate or citric acid. So watch me as we draw the pathway or the cycle together. Okay? The cycle starts from the entrance of the pyruvate. The pyruvate into the mitochondria because that's where the TCA cycle takes place. So one of the fates of a pyruvate is that the pyruvate is converted to acetyl-CoA. Acetyl-CoA. By an enzyme called pyruvate dehydrogenase complex. And this is a multi-enzyme complex that contains 3 enzymes and 5 coenzymes. The 5 coenzymes / cofactors are NADH, NAD plus, FAD plus, TPP, which is thymine pyrophosphate lipoate, and CoH. So it contains 5 coenzymes / cofactors and 3 enzymes. The first enzyme is E1, which stands for pyruvate dehydrogenase. The second enzyme is E2, which codes for dihydrolipoid transacetylase. The third enzyme is E3, which stands for dihydrolipoid dehydrogenase. So pyruvate enters the mitochondria and is converted to acetyl-CoA. And the first reactions of this cycle is that this acetyl-CoA condenses with a molecule called oxaloacetate to form citrate. So as you can see, citrate is the first molecule that is formed, and it contains 3 molecules of carboxylic acid. That's why it's called the TCA cycle. And it's called CAC cycle because this is the first molecule or first product that is formed. So condensation. Step 1 is the condensation of acetyl-CoA with oxaloacetate to form citrate by an enzyme called citrate synthase. So the CoA will leave as CoH. So remember, it's condensation. Water is also required. So this is step 1. Step 2 converts citrate to isocitrate. So this step 2, we have 2A. 2A, which involves the dehydration of citrate to what we call cis-aconitate. 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 So this is 2A. This is step 1. Remember, it's dehydration, removal of water. So 2B is now the rehydration. Rehydration. Addition of water to cis-aconitate to produce isocitrate. So this is 2B. And the enzyme is aconitase. Aconitase. So step 2 is the conversion of citrate to isocitrate by an enzyme called aconitase. Step 3 is oxidative decarboxylation of isocitrate to alpha ketoglutarate. So alpha ketoglutarate. Oxidative decarboxylation. So CO2 is released. So this reaction requires one molecule of NAD plus to produce NADH. And the enzyme is isocitrate dehydrogenase. So this is step 3. Step 3. Step 4 is the oxidative decarboxylation of alpha ketoglutarate to produce succinyl CoA. Succinyl CoA. So we also have the same thing that happened here. One molecule of NAD plus produces NADH. So and the enzyme is alpha keto dehydrogenase complex. The difference between this complex enzyme and this one, PDH complex, is just E1. So this one has, both of them have 5 coenzymes slash cofactors and 3 enzymes. So still E1, E2, E3. The difference is that this one, E1 of KDH complex, codes for alpha ketoglutarate dehydrogenase, while E1 of PDH complex codes for pyruvate dehydrogenase. So step 5 is the conversion of succinyl CoA to succinate. So what we call substrate level phosphorylation, it takes place at step 5. To succinate. So we have ADP plus pi to produce ATP. Meanwhile, we can also use GDP plus pi. Pi is inorganic phosphate. So the enzyme is succinyl-CoA synthetase. Succinyl-CoA synthetase. Synthetase. So step 6 is a conversion of succinate to fumarate by dehydrogenation. By dehydrogenation. So this one uses FAD plus to produce FADH2. Meanwhile, the intermediate formed between succinyl-CoA and succinate, we call it succinate phosphate. So step 6 is the dehydrogenation of succinate to form what? Fumarate. Step 7 is the hydration. Okay, the enzyme here is succinate dehydrogenase. Succinate dehydrogenase. Step 7 is the conversion of your fumarate to malate. To malate. To malate. The instructions are simple. Just have to learn it. So hydration is the addition of water, right? And the enzyme is fumarase. Fumarase. Fumarase is the name of the enzyme. Fumarase is the name of the enzyme. So lastly, malate is dehydrogenated to form oxaloacetate. So making use of NAD plus. So this NAD plus, actually look at what it did. Collected these two hydrogen here to form NADH plus H plus. And the enzyme is malate dehydrogenase. So you have step 1, step 2, step 3, the conversion of isostrate to alpha ketoglutarate by IDH, isocitrate dehydrogenase. Step 4, the conversion of alpha Kg to succinyl-CoA by alpha KDH complex. Step 5, the conversion of succinyl-CoA to succinate by succinyl-CoA synthetase. Step 6, the conversion of succinate to fumarate by succinate dehydrogenase. Step 7, the conversion of fumarate to malate by fumarase. Then last step, which is step 8, is the conversion of malate to oxaloacetate. Now let me talk about the reactions. So step 1 is condensation reaction. Step 2 is dehydration and rehydration. Step 3 and step 4 is oxidative decarboxylation. So the oxidative decarboxylation reactions of the TCA cycle are steps 3 and step 4. The irreversible steps of this pathway, look at it. Step 1, the ones with a single line. Step 3 and step 4. Other steps are reversible. So we can also call the irreversible steps the regulatory steps. Okay? The oxidation reactions of this cycle is step 3, step 4, step 6, and step 8. Step 6 and step 8 is dehydrogenation reactions. And what's dehydrogenation? Removal of hydrogen. And removal of hydrogen is called oxidation. Also, now look at this. Let's look at the bioenergetics of this cycle. The cycle, the places where ATP or high energy carriers are produced. Now you can see step 3. Step 3 produce one molecule of NADH. Step 4 produce one molecule of NADH. Step 5 produce one molecule of ATP. Step 6 produce one molecule of FADH2. Step 8 produce one molecule of NADH. And NADH is equivalent to approximately 3 ATP in the electron transport chain. We have 3 ATP, while FADH2 is equivalent to approximately 2 ATP in the electron transport chain. So we have this 2 ATP, then 3 ATP. So total is what? 15. Okay, 12 rather. 12 ATP. So 12 molecules of ATP is produced from the oxidation of one molecule of acetyl-CoA in the TCA cycle. So this is the cycle, is the end of the lecture. Do well to share, like, and comment. Have a wonderful day.